Hello everyone, I am Dler and welcome to Biology. In today's video, we are going to talk about DNA replication. DNA is a molecule made up of two strands twisted around each other in a double helix shape. DNA replication is the basis of inheritance. It occurs in all living organisms, acting as the most essential part for biological inheritance. DNA replication is a process by which DNA is copied during S phase in a cell before a cell divides by mitosis, meiosis, or binary fission. During replication, the two strands of DNA separate from each other. Each strand serves as a template for creating a complementary strand. After replication, the two identical double-stranded molecules are then separated and moved to a new cell formed during cell division. Before the nature of DNA replication was known, there were three putative hypotheses that could describe the nature of DNA replication process. The first one, the outer strand is created in a completely new manner, while retaining the structure of original parent strand. This is the conservative model for DNA replication. The second one is dispersive model, where the parent strand would be interspersed with the newly replicated sequence of DNA. The third one is semi-conservative, where the resulting product each contains one daughter strand and one parent strand. The semi-conservative nature of DNA replication was discovered through a labeling experiment using different isotopes of nitrogen. In the first part of the experiment, the entire parental DNA is labeled with 15 nitrogen or heavy nitrogen. The culture is then shifted and grown into 14 nitrogen, which is the light nitrogen. If DNA replication is conservative, all of the heavy nitrogen would stay together, and the lighter nitrogen would also stay together. But if DNA replicated in a semi-conservative or dispersive model, after one round of replication, it would create a single band of DNA that has mixed with 15 and 14 nitrogen mixed together. However, after one round of replication, you wouldn't be able to tell the differences between semi-conservative or dispersive model. For that, you're just gonna have to do a second round of replication. In this case, semi-conservative would result in a product that would either have a 14 and 15 nitrogen mixed together or be completely 14, while dispersive model would still have a single band of DNA that has mixed with 15 and 14 nitrogen. So what was observed is the semi-conservative predictions of single band of DNA after one round of replication and two band of DNA after second round of replication. This was the first indication that parent DNA acts as templates for creating daughter strands. So the first step in DNA replication is to separate the two strands. This unzipping is done by an enzyme called helicase. Helicases move along the DNA molecules, breaking the hydrogen bonds between the complementary nitrogenous bases. This action allows the two strands of the double helix to separate from each other. The Y-shaped region that results when the two strands separate is called a replication fork. Once the two strands have been separated, they run in opposite direction. This determines how each strand of DNA is replicated. One of these strands moves along with the replication fork, which we call it the leading strand or continuous strand, while the other strand runs in the opposite direction, away from the replication fork, and we call it the lagging strand or discontinuous strand. An enzyme called primase starts the process. This enzyme makes small pieces of RNA called primer. In the leading strand, we only need one primer for the construction of the new strand of DNA. At the 5' end, Then another enzyme, DNA polymerase, comes into play and binds to the primer and will make the new strand of DNA. DNA polymerase can only add DNA bases in one direction, from the 5' end to 3' end. But the lagging strand, it cannot be made in this continuous way because it runs in the opposite direction. So DNA polymerase can therefore only make this strand in a series of small chunks called Okazaki fragment. Each fragment was an RNA primer. Then DNA polymerase added a short row of DNA bases in the 5' end to 3' end direction. And the process is then repeated and more Okazaki fragment will be made. And then we have SSBP, which stands for single strand binding protein, prevents the two strands from cleavage and snapping back together. As helicase separate the two strands, torsional strain is created ahead of the replication fork, so an enzyme is needed. DNA gyrase, which is a type of topoisomerase, reduces the torsional strain and even relieves positive supercoiling that could form in the DNA strand ahead of the replication fork. Once the new DNA has been made, the enzyme exonuclease removes all the RNA primers from both strands of DNA. Another DNA polymerase then fills in the gaps that are left behind with DNA bases. Finally, the enzyme DNA ligase seals up the fragments of DNA in both strands to form a continuous double strand. As I said before, DNA replication is described as semi-conservative because each DNA molecule is made up of one old conserved strand of DNA and one new one.